All right, I got a red light.
Yep, now it's recording.
Okay, awesome. All right, so I'm going to go over chapter, I think this is 15 and 16. So today I'm going to go over the adaptive immune system, which is going to be a little bit complicated for everyone if you've never had this before, or maybe it's review, or maybe a little bit more detail for you. So I don't know, in the past, maybe they told you B cells make antibodies, or T cells fight cells directly. Maybe that was the extent you got. I'm going to go into a little bit more detail talking about antigen presenting cells. And I know I mentioned before that these are the dendritic cells, your monocytes, macrophages, and your neutrophils, which are going to actually capture antigenic particles that are foreign to your body, process them, and present them to your immune system. All right, so your immune system, once it recognizes as foreign, if B cells match, that lock and key mechanism matches, they will start producing antibodies to that antigen. All right, we'll present it to a CD4 helper cell. And I said before, that those are the ones that are going to triage your entire immune system. I mentioned that on Tuesday, I think, right? So your CD4 cells are going to triage. They're going to tell your B cells, this is the antigen. You recognize it, the B cell agrees, all right? They, their markers find each other, their receptors find each other, they agree, they handshake, all right? They sign a little contract, and your B cells start making memory cells for future, all right? They make clones of themselves, so we have more, all right? And your CD4 cell is going to initiate activity with your CD8 killer cells. And I mentioned the other day that your T cells, remember, these are hand-to-hand combat. They'll actually go in. These are cell-destroying cells, all right? B cells work with antibodies. Those are going to be blood-borne and in your tissues and in your lymph nodes. So they don't actually kill anything. They actually attach and opsonicize, neutralize, or super tag the antigen for your immune system so that it's easier for your T cells to find it, other macrophages and monocytes to find the cells and initiate whatever they can do for it, right? So this is your third line defense. This is acquired. So remember, this, you're not born with this. We talked about innate on Tuesday. You're kind of born with that, and it's kind of a genetic system. And we mentioned pathogen molecular patterns, all right? So your innate system will recognize things that are relatively foreign or similar as foreign, and they'll initiate a basic response. This is going to be very specific, acquired, and I said before, this is kind of the basis for your vaccinations, that you're presenting these antigens, these antigenic particles, either in full and, you know, they're going to attenuate them, kill them, or give you parts of it, so that your immune system, your dendritic cells will grab those and process them and present them to your immune system so they can specifically make antibodies to it. So if the antigen is dead or attenuated, or just a part of a capsule or part of a viral particle, all right, the theory is that it's going to initiate that immune response. So we have antibodies to it, and our T cells are notified, and we can have a much quicker response should we actually come in contact with a live or pathogenic strain of bacteria or viruses. All right, does that make sense to everybody? Maybe? All right, you can watch it later. All right, so there's two features that characterize specific immunity. So specificity. Antibodies produced function only against the antigen they are produced in response to. And we're going to see later, your B cells, they said we're going to shuffle all your genetics, all right, and make a bunch of B cells to make all these different combinations, like a quick pick in the lottery, and then they're going to sit there naive or unactivated until they come across the antibody that matches that code. Once they do, the B cell is no longer naive. It's activated. It'll start producing antibodies. And then we said before, or I said on Tuesday, we want memory cells. So your lymphocytes are programmed to recall their first encounter with the antigen and respond rapidly to subsequent encounters. So if you've been vaccinated, let me just check and see if I can admit Abby. Okay. So with vaccination, we want to program those B cells and the T cells to that antigen. So if we do actually come across it, we're able to have full defense up and running within 24 to 48 hours. It's already there. We don't have to wait to activate B cells, and we have to wait that 73-hour to 5-day process to get our titers up in our blood. And our T cells are already programmed. Once they find the antigen marker on something foreign, they go right in and go for the kill. Right? So these are separate but related activities of the specific immune system. Right? They develop and differentiation of the immune system. So we have to activate them. They're going to develop and they're going to differentiate to a very specific antigen or antigenic particle. Right? And we have our lymphocytes and antigen processing, and that, remember, that is done with your dendritic cells, right? Or your they're called antigen presenting cells. All right, we're gonna find out if we have MHC2 markers, all right, and they're going to be markers, and I want you to remember they're presenting it. So just think of it like two hands presenting the antigen to your immune system. Okay, cooperation between the lymphocytes during antigen presentation. So our dendritic cells or antigen presenting cells are going to present these to our lymphocytes. And remember, our lymphocytes are those CD4 or CD8 cells, all right, and our B cells. These are our lymphocytes. So we're presenting it to them, and they're going to agree they're antigenic or foreign, and they're going to initiate the immune response very specifically to that antigen. B lymphocytes and the production and actions of antibodies. Once again, B cells, lymphocytes produce antibodies. These are in the blood. And once again, B cells produce antibodies through plasma cells. Remember I said they're going to probably try to trick you on a board exam, MCATs, OCATs, whatever you're taking. And then we have our T lymphocyte responses. So T lymphocytes are your CD4 helper cells, the triage cells, and our CD8, which are our killer cells. They directly kill the cells. And then we're going to have something called natural killer cells we'll talk about briefly. They're also part of those lymphocytes from that bloodline. Remember, we have those two bloodlines. This is the lymphocytic bloodline. Right. So here's a chart. I don't know. I reviewed it earlier. I don't know if it's that clear. All right. But it's just telling you here, these are your B cells. So these are produced in the bone marrow. And these literally go into your bloodstream, fully developed. Right? And they're, like I said, the B cells are just waiting for the antigen that matches their specific random code. Right? And your T cells, I'm sure you've learned before, go to the thymus, and they have to remember, they have to do two things I mentioned the other day. They have to figure out whether they're CD4 helper or going to be a CD8. They have to declare their major, right? And remember, the other thing I mentioned is they have to be able to recognize self from non-self. So if we have one of these, that CD4 or CD8 that has a reaction with self cells, we want that clonal deletion from Tuesday's class. I said we don't want them to replicate themselves because they're faulty. We don't want these cells running around your body, attacking your own cells, thinking that some component of that is foreign when it's actually part of you. And this is going to be the basis for part of chapter 16 about your autoimmune diseases. We'll go into like sequestered theory and other things briefly in next chapter. I won't get into a lot about autoimmune diseases, but just some basics for your board exams or NCLEX or whatever you're taking. Anybody else is joining us? Okay. All right. So the second part here, this is just an overview, and we'll go over the specifics. So contact with antigens, all right, we have presentation by antigen presenting cells, dendritic cells, monocytes, macrophages, and your neutrophils are also all part of cells that can recognize these foreign particles, and they present them to your immune system. Foreign cells bear molecules or antigens that are recognized and engulfed by dendritic cells and macrophages or B cells. For most responses, specialized cells called T helper cells, those are your CD4 cells, right? First receive the processed antigen from the phagocyte, right? That just means a cell that consumed it. Right? Those antigen-presenting cells process the antigen, all right, and they're going to stimulate the B or T cells. Remember, they're going to present these on their MHC2 markers. And then we want the overall response we want to activate B cells to produce an antibody to that specific antigen. And remember, these are naive. They don't produce antibodies until these antigen-presenting cells present it to the B cell, and the B cell says, all right, I've been waiting my whole life to find the actual antigen that will trigger my cell to produce antibodies, right? So they're all naive when they leave the bone marrow. We set up here, they're fully mature, but they're naive, all right? So they're little virgin B cells. They haven't been shown their magic lock and key mechanism. Once they get that, they start making antibodies through their plasma cells, and they're gonna make, right here, they're gonna make memory cells, and we want these around. So if we ever come across it again in the future, we automatically have these activated cells. They have the magic recipe for the antibody, and they're going to reproduce the antibodies in great amounts. And the other cool thing about this is once you have memory cells that get activated, they stick around for a very long time. They want to make sure that there's nothing around that they haven't put an antibody. They don't want any antigens floating around that aren't coated with an antibody. So we can make these great big molecules, and your T cells really can't miss it. We have these antigens on them. The T cells come along, they see that marker on the MHC1 markers, and they go in and attack. And remember, they literally rip a hole in whatever it is, and they send in chemicals or lysosomes to destroy it. Everybody lost yet? Okay. All right, so we have cell receptors or markers confer specificity and identification of a cell. So the major functions of the receptors are, right, so these are receptors on your cells, all right, and they're, they can't really scan. There's receptors that are going to find those pathogen-associated molecular markers in your innate system, or they're going to find specific MHC1 or HLA markers on anything that's antigenic and recognize it. So, let's say that you had some kind of bone marrow transplant or organ transplant. Remember, those things are not part of you. So they're MHC1 markers, also known as your human HLA, human lymphatic component complexes, are going to be different. All of you, unless you have a twin out there, all of your MHC1 or HLA markers are all specific to you. So this is what these cells are going to, anything that it isn't part of your MHC1 or HLA markers is going to hopefully stimulate your immune system. Right. To promote the recognition of self molecules, we want to make sure that your immune system, when it's bumping into its own cells, recognizes it as self and doesn't attack it. And when we start talking about the bacteria and some of these other molecules that actually jump inside of your macrophages and monocytes, remember, they're in there replicating. Your immune system says that's a self molecule. They're not going to attack it. So that's a big virulence factor for these bacteria. They can literally hide out in your antigen presenting cells or monocytes. And I said before, they get a free Uber ride anywhere they want. No one's going to bother them. They can literally just leave the cell, or they can cause the cell to burst. They've replicated inside your cell, and now they have a great place a little womb to grow in, and they can just leave anytime they want. All right. They receive and transmit chemical messages among other cells of the system. So they're going to send out those cytokines to say, I need more help here, or I'm going to send out some interferon. All right. I'm going to stimulate more B cells to produce more antibodies, or I need more memory cells, or they're going to send out signals directing the immune system on what to do. And they aid in cellular development. Like I said, with these chemicals, they're going to tell the immune system, I need more help. I need more antigen presenting cells, or I need more B cells, I need more antibodies, or I need some more T cells. So they're going to direct whatever's going on. And I mentioned on Tuesday, if you have a problem with your CD4 triage cells, those helper cells, your immune system can't do a lot of these things. So your immune system comes to almost a standstill. And, you know, we say people die from HIV or these immune compromising diseases, but literally, technically, they die from complications of it. They don't actually die from the virus itself. They die from the virus hijacking all the CD4 helper cells, and you've got nothing to triage your immune system. Right? So I know I mentioned MHC 1 markers, all right, and these are the same as your HLA markers. So they're going to use these two terms interchangeably. They're the same thing. So these receptors are found on all cells except red blood cells, all right? And that theoretically is because red blood cells don't have a nucleus, right? That's the exception. They're also known as human leukocyte antigen. Leuko, white, cyte is the adult cell. So these are human white blood cell antigens, HLA markers. All right, so if you have an organ transplant or you have a bone marrow transplant, for that transplant, we want to make sure that these HLA markers are as close to yours as possible to reduce the amount of rejection from your system. We don't want to really irritate and stimulate your immune system to attack the bone marrow or the organ that we're implanting for. So it plays a role in recognition of self by the immune system and in rejection of foreign tissue. All right, so I want to make a key point here, so when you go back and review this, you can make notes or whatever. Class 1 MHC molecule is found in all nucleated human cells. So every cell that you have in your body, or if you have an identical twin, every cell has an MHC 1 molecule. Right? So those are the 1 molecules, same as the human lymphocyte antigen markers, same exact thing. Right? Now, the other thing is, I said we had antigen presenting cells, and I said I want you to remember those as presenting something. So these MHC2 markers are found on some types of white blood cells. Right? And when they say some types, this is your antigen presenting cells, the ones that literally your leukocytes that go in, recognize antigens as foreign, they process the antigen and present it to your immune system. Right? So these functions of the MHC markers. So the class 1 markers that display unique characteristics of self molecules and regulate the immune reaction. All right. So, like I said, these are the same. Every one of you has your own specific class 1 markers on all of your cells that are nucleated. Red blood cells are the exception. And these are required for lymphocytes. And what that means is, I said before that the T lymphocytes, your CD8 lymphocytes, are your killer cells. When they find a class 1 marker of something that isn't you, so an organ transplant or bone marrow transplant or some kind of transplant or any kind of foreign particle, whether it's a bacteria or whatever, if it has a class 1 MHC marker that's foreign and your immune system has already recognized it, this is where they attach. They know it's foreign, they attach it, and this is where they have the hand-to-hand combat and kill it. Your class 2, remember these are the presenting ones. Regulatory receptors found on macrophages, dendritic cells, and B cells, and neutrophils, but your book doesn't get into that, but anyway, they can do that. These are your antigen presenting cells, right, involved in presentation of antigens to your T cells. And by T cells right here, we mean CD4 or CD8. So lymphocytes' role in surveillance and recognition is a function of their receptors. That's how they process everything. So they don't have Wi-Fi scanning codes or whatever. These are done by their receptors. So B cell receptors, these bind free antigens. So B cells are your plasma cells. Your B cells are your plasma cells. These bind free antigens. So if you have antigens floating around, all right, and these B cells have been activated to this antigen, so they were naive. Once they're exposed to the antigen, right? They start making antibodies. So when they see it again, they're going to bind to these free antigens, right? And like I said, they super tag it by opsonization. They go in and they tag it so that your macrophages and monocytes can't possibly miss it, right? They're going to grab it and process it again, and they're going to kill it, T cells. All right, T cell receptors, these bind process antigens together with the MHC molecules on the cells that present antigens to them. You'll see that coming up here. So lymphocytes use 500 genes to produce tremendous varieties of specific receptors. All right, so this is the clonal selection theory. So this is absolutely, to me, it's amazing. So your body is going to take 500 genes. They're going to make tremendous amount of quick pick shuffles, and they're going to make all these B cells, all these tremendous combinations, and it makes all these B cells in your bone marrow, sends them out. And remember, these are naive. They have no idea what they're going to come across, but we kind of make millions of combinations. So if they come across any antigen, known or unknown, they're going to produce antibodies to it. So it's really amazing. Undifferentiated lymphocytes undergo a continuous series of divisions and genetic changes that generate millions of different cell types. So we're going to take these 500 genes, we're going to shuffle them all around and make millions of combinations of B cells that are naive, and they're just waiting for that specific antigen that matches that code. Right? Each cell has a particular unique receptor specificity. Remember, these B cells can only respond to one single antigen. So it's pretty amazing. The other cool thing that we'll see coming up here is that you can have an antigenic particle or something, and it could have 5 or 6 different antigens on its epigenetic, and you could literally have 4 or 5 or 6 B cells that attach, and each one's going to make a unique antibody to that specific antigen. So we could theoretically have 5 or 6 completely different antibodies attaching to it, and we super tag this thing, and your immune system, if it's up and functioning, should be able to respond to that very, very quickly and very, very effectively. All right. So the development of lymphocytes, I'm hoping this is a little bit a review for you. In the bone marrow, lymphocyte stem cells differentiate into T or B cells. So we know your bone marrow makes all of your blood cells. So it's going to be B or T. Right? Remember, your B are in your plasma. They make antibodies. Your T cells have to go to the thymus, and they have to decide CD4, CD8, and they have to be able to tell self from non-self. If they can't tell self from non-self, remember, that's clonal deletion. We want to just get rid of that. We don't want defective T cells floating around. Very bad. B cells stay in the bone marrow while T cells migrate to the thymus. Both T and B cells migrate to secondary lymphoid tissue. Right? And we talked about that on Tuesday. Remember, your secondary lymphoid tissue is all of your lymph nodes, your Peyer's patches, that MALT, and the GALT, the GI-associated lymphatic tissue, or anywhere, anywhere in your body that is, remember, these are in any part of your body that's exposed to the outside. And if you're not sure what that is, that's going to be your skin, or anything that is lined with the mucous membrane is literally outside of your body. So here's the really cool part. All right, so lymphocyte specificity is pre-programmed, existing in the genetic makeup before an antigen has ever entered the system. So what they're saying is these B cells, we make millions of combinations, and they're specific for a certain antigen that you may never come across, and it's never even entered your system yet. So depending on your genetics, we mentioned the other day that genetically some people are more susceptible to disease and some people have super strong immune systems. So genetically, however, whatever genetics you came in with from your parents or distant relatives or your bloodline or however you want to describe it, that's going to be your genetic stock. Right? So your body's going to go in and it's going to shuffle those genes randomly, all right, and make all of these super naive B cells. And remember, each B cell is going to be specific to a certain antigen. Each genetically different type of lymphocyte, so these are your clones, expresses a single specificity for one single antigen. It's just kind of showing you here simplistically. Here's your lymphocyte. Here's your stem cell. It's going to make all these different B cells. And, you know, theoretically, these are all supposed to be slightly different. All right. And then these are all going to be very, very specific different antigens. And remember, there's millions of these, completely random combinations. All right. The problem here is it's showing you if you happen to have a B cell that locks on itself as a self tissue. Oh, okay, sorry. Hold on. Okay, guys, sorry. Oh, I screwed up something. Allow participants to unmute themselves. Sorry. I hear something. I'm so sorry, guys. I think I messed up something. Okay. Okay. All right, does anybody have a question before I screw up more? No? Good for it. Okay. So, oh yeah, so here we're seeing a B cell that recognizes, it thinks self is foreign. So we want to eliminate these. So it's going to be called clonal deletion. We don't want that replicating. We don't want a bad B cell replicating. So clonal selection, first introduction of each type of antigen into the immune system selects a genetically distinct lymphocyte. Right. Causes it to expand into a clone of cells that can react to that antigen. So once we get a specific antigen that we've never been exposed to before, it's going to trigger a B cell, probably in your interstitial tissue, hopefully not your bloodstream, or where it really happens is your lymph nodes. You know, we talked about your lymph nodes pick up all that extracellular interstitial fluid, your lymph vessels, and they bring it to your lymph nodes. And we saw the afferent and efferent channels. All right, we want that to percolate through your lymph nodes, and we want those B cells to percolate to get exposed to the antigen. And we really want that antigen to trigger a B cell. The B cell's triggered. All right, it starts pumping out. It's waited its whole life to get its receptor triggered. It starts pumping up antibodies. All right, we're going to signal that nucleus to make antibodies. All right, and then it's going to signal other B cells to make plasma cells. So we have the recipe, and we can start pumping up more and more antibodies. And remember, all it takes is one B cell to get activated. We start making antibodies, and we start making memory cells. So it's pretty amazing. All right. So receptors, genes of B cells govern the immunoglobulin Ig synthesis. So you guys have probably heard of IgA, IgD, IgE, and IgM. All right. So these are immunoglobulins, and when we talk about blood titers, these are the things that are elevated in your blood. So if your blood titer for a specific antibody is elevated, we have an idea of what's going on in your body as far as infection or what might be causing a problem. All right, so these are your light chains up here. I'm hoping this is review. It should, I think this was in your chemistry packet. If not, I think it's in the immunity coloring book thing that I uploaded for Blackboard. So these are your light chains, this is your heavy chain. So this part down here, the darker, I don't know if it's green or purple, the heavy chain is the same for all of your antibodies, all right? The light chain is where the antibodies bind to, right? The variable region, V stands for variable region. So antibodies actually attach to this very specific lock and key mechanism, And then once it locks there, it's going to send a signal here. This is attached to your B cell. It's going to send the B cell a signal. The B cell starts making the antibodies. Okay. Let me just check. Allow, just missed. Hold on. Mute.
Says you're trying to unmute me. Was that on purpose or?
Yeah, because I was told that you guys can't talk. Was that Anthony?
Yeah, that's me. That's me. I just got an alert on my screen saying you were trying to unmute me. I had it.
Because Robert's telling me that you guys can't, it won't let you guys speak. So I can mute you again. I'm sorry.
I'm good.
What the hell is going on with this? Yeah, I'm just afraid somebody may have a question. That's my problem.
There we go.
Yeah, this is so weird.
Okay, you got me unmuted.
Just ask everyone to unmute in case they have a question. It's terrible.
Can anybody else unmute?
I don't know what's going on. It should say
I can unmute. Can you hear this?
Yep.
yes.
Yep, I'm unmuted now.
How is going on with this thing?
Anthony, are you still good?
He muted himself again.
Oh, okay.
All right, let me.
Technology's a pain in the butt.
Oh, it's me.
No, he's saying he still can't talk now. It's like it's picking and choosing who it wants to unmute.
No, I clicked unmute myself.
Oh, okay.
All right, no, I'm sorry. I clicked mute myself and then wasn't able to turn it back. I was just trying it out, see what happened. So
Looks like Kathleen can't unmute.
There, she's unmuted. Catherine.
Michelle as well.
it's just some setting, I suppose.
Okay, I'm unmuted.
I hit unmute. It's like, yeah, I could literally hit it.
I felt bad. You'd ask, do you have any questions? I'm like, I can't say anything.
All right. While I have some actions and maybe questions.
I don't think so.
I had one, but I forgot it. So sorry.
No, don't apologize. I don't know clearly. Let me back up here and make sure that got to this. I can't even swear because I'm recording this right now.
That's all right. If it's any consolation, this is what my job's been like this whole time too. Just
okay, now we can talk.
New streaming services and screen sharing and stuff never seems to work for us.
Okay.
So, all right, if you guys have a question, you guys can, why don't you just text it to me if you guys want to write down my number.
Can you open up the chat thing that we can type into as well? Because
Nope. All right, let me try that.
There's a chat window at the bottom.
Yeah.
Yeah, at the very
if
bottom.
you drop your mouse to the bottom, you should see next to participants thing that says chat. Oh. No, Daddy's in class, honey. Go play. Wait, can I have some ice cream?
Oh, I got everyone's picture. Show after speaker. You sure it's at the bottom?
Mine's at the bottom. Yeah.
You guys can see my mouse, right?
Yep.
Will you leave these? You aren't supposed to.
I'm afraid to unshare my screen. Hold on, let's see chat. I don't know this is unbelievable. Zoom group chat. It seems to be open.
That's what we've been using to just kind of write each other.
This isn't. Everyone publicly. Did that work? I feel like I'm two. I can't figure this out. All right, I have nothing come open on the bottom. I swear to you, nothing. Either my keypad or my magic mouse. Nothing is going on. All right. Show active speaker. Show greater video. Chat.
Can you put the milk in the fridge? We got these. Look at.
Okay, I give up, guys. I don't know what to do here.
What is it?
The security tag.
Request the remote control. Let's see if I can. I can request the remote control. All right. Okay.
Pull that one forward. I'm
going to pass the baton to Robert. Okay, that helped.
There's the chat.
Where?
I just popped it up.
Where? Oh,
Could you? Is it just on my screen or is it on yours?
I just have something up right now because I just lost the control bar at the bottom. All right. I guess, yeah. Control again. I don't know what else to do.
Let me give it back. Hold on. There. I just gave it back to you.
I don't want it.
Okay. All it does is access the shared content for the remote. I thought at least I could like change the settings on it, but it won't let me.
I swear to you, I have no idea. Maybe we'll have to go back to WebEx, unfortunately. But, all right. Okay. Let me get through this. We did this already. Okay. All right. So basically, these are your antibodies, and everyone's heard of this, right? So we have two identical heavy chains. Remember, I said these don't change. These are on every single antibody. The variable region here is what makes each single antigen part of each B cell specific for each antigen. And remember, there's millions of combinations on different B cells. And this is where your antigens, your foreign particles of whatever it is, are going to attach and trigger this B cell to produce antibodies. Right? So those are your antigen binding sites. Right? Variable region is the V region. Everything else is constant. There's also known as your heavy chain. All right, they can ask you that somewhere. All right, these are sulfur bonds if anyone cares. Disulfide bonds if anyone cares about that. Right. So immunoglobulin genes lie on 3 different chromosomes. Like I said, I'm not going to ask you guys, you know, how we jumble these genes. All right, but just realize they have 150 genomes, and we can make millions of combinations by just randomly picking our V region, our D region, our J region. We shuffle them all around, and we can genetically make millions of different B cells with very specific antigen receptors, which is really amazing. So, you know, that's why you can come across anything in the world theoretically and produce an antibody to it. It's absolutely amazing. Right. So once synthesized, immunoglobulin is transported to the cell membrane, is inserted there into there to act as a receptor. So once your B cells make it, we actually take this particle here and we insert it onto all the new B cells. So whenever they come across that again, they automatically have the recipe, and they'll literally, once they're triggered, they're going to pump out a whole bunch of antibodies, right? So if anyone's heard of anaphylactic shock, all right, or these over hypersensitivities, this is what happens on some of your eosinophils, right? They are locked and loaded. So the minute anything irritates it, they just start pumping out a bunch of histamine and things, and cause a major reaction. Very, very similar to that with your basophils or your mast cells. Remember, your basophils are in your blood, your mast cells are in your tissue. Right? And by tissue, I mean your epidermis or that mucous membrane. Right? Anything that's exposed to the outside surface, these mast cells are going to hide out there. They're not going to be in your blood. They're going to be out in your tissues, just waiting for any kind of foreign antigen to stimulate them. All right, now, we were just talking about B cell receptors and your antibodies and that variable region and that heavy region. Now we're going to get into the T cell receptors, okay? These are formed by genetic recombination with variable and constant regions, just like the B cells, right? But their components are a little bit different. These have 2 parallel polypeptide chains. All right, they still have the 2 chains, but they're just shaped a little bit different. They're variable regions in here. This is their constant region. Right? These are small, but these are not secreted. Remember, your B cells, once they're activated, we actually secrete these antibodies, and they go and attach to that B cell. All right, so B cell maturation. So you guys probably all heard, and by guys, I don't mean men only. I know there's women in the group, but that's just kind of a general colloquialism. All right, they're directed by bone marrow sites that harbor stromal cells. Now, if you don't know what stromal cells are, these are cells in your bone marrow or your lymph nodes or other parts of your body. If you ever hear stromal or stroma, these are cells that actually are, I don't want to make the term like nannies. They actually go in and they nurture and they feed these cells chemical signals to help them mature or grow or replicate. All right, so they're kind of nannies or babysitters or whatever you want to use. All right, and they provide hormonal signals. So millions of distinct B cells develop, and they actually go to, we said before, there's specific sites in the lymph nodes, spleen, and gastrointestinal associated lymphatic tissue, all right, or MALT or whatever. So any of your secondary lymph sites, which are going to harbor all of your B cells. And we want those B cells there as things are percolating through your body or your lymph vessels. We want them to go to your lymph nodes. We want them to go to your spleen. Anything in your digestive tract, we want it to go to here so we can, any antibodies, or I'm sorry, any antigens that you consume or swallow, or any foreign particles that invade your body through your digestive tract or you swallow it, we want to trigger these B cells, and we want to trigger them right away. We don't want them to get into your blood supply and interstitial fluid to trigger. We want to have the sentinel right there. We want those things to activate lymph tissue there. So if we have a B cell that's already been triggered, it's there, or if we have a naive B cell that will recognize this foreign particle that you consumed, we can wage the response immediately. All right. The one that's missing here are your tonsils. Everybody have their tonsils or have them taken out? All right. Your tonsils, I want you to think of those as kind of like flypaper or whatever. So when you're breathing something in or swallowing it, you have that lymphatic tissue in the back of your throat. If we can trigger that immediately before, as you swallow it or whatever, before it gets into your digestive tract, we can trigger that B cell response as soon as possible. They come into contact with antigens throughout your life. They have immunoglobulin as surface receptors for antigens. So we want these B cells, we want them all over your body, anywhere that's exposed to outside, and we want to trigger this throughout your life. We want to trigger it as soon as possible. Right? Now your T cells, I'm sure you learned, maturation is directed by the thymus gland and its hormones. And I said before, these T cells have to determine whether they want to be CD4 or CD8 cells. And CD means cluster of differentiation. So we're saying the T cell goes to the thymus, it has to declare its major. It has to say, well, I want to be a CD4 helper cell, or I want to be a CD8 killer cell. And we'll talk about natural killer cells too, which are the other division. All right. The other thing, before I forget, please realize you have some CD8 cells that are regulatory, all right? And their job is to, if your immune system goes out of control, we have CD cells that are kind of like police. They kind of sedate or put the brakes on your immune system. The book doesn't talk a lot about them. It's out of the scope of this course, but please realize you have other CD8 cells along with the natural killer cells that literally they police the area, and they kind of tell some of your CD8 cells if they go crazy to calm down a little bit. And we'll be talking about Staph aureus and some of these other bacteria that secrete factors that make your CD8 cells go absolutely bonkers. All right, so your CD8 cells, they trigger all your CD8 cells to go ballistic and attack anything they come in contact with. All right, so it's a huge virulence factor for Staph aureus. It causes a major inflammatory and immune response, causes a lot of havoc, and keeps your immune system busy. The Staph aureus has chances to just invade relentlessly. Mature T cells migrate to lymphoid organs. All right, and once again, they're going to hang out with the B cells. All right, they're going to play video games or whatever they do, just waiting for that specific antigen to come along. Right. So here's just a chart. Like I said, I don't know if charts help you guys or not. It always helped me kind of figure out the differences between things. So B cells, their site of maturation, where do they mature? Their markers we talked about. All right. B cells circulate in your blood in very low numbers. All right. T cells circulate in very high numbers. All right. And if you don't believe me, go through your blood counts again. We talked about CBC with differential. Remember, never let monkeys eat bananas. Go through and figure out the percentage in your blood. Your circulating in your blood, B cells are in low number. Your lymphocytes are much higher. All right. So it would be the second highest number. Receptors for antigens. All right. On your B cells, it's usually it's D or M. Now, one of these is going to be very, very big. We'll see coming up. It actually is a pentamer. It's got 5. It's humongous. All right. All right. I don't really care, about the distribution of the lymphoid organs. All right. I don't know if you guys know about the cortex and the follicles. That's more of a pathophysiology question. All right. B cells, remember, these do not require antigen presentation to be activated. Right. So they'll find them on their own. T cell, these are very, very effective, but they literally need a formal introduction from a CD4 cell. These are natural born killer cells, but they actually have to be formally introduced by a CD4 cell. All right. All right, plasma cells and memory cells produce the antigen stimulation, right? And then general functions. So kind of remember your B cells are always going to be bloodborne, and they're going to be really using antibodies to inactivate, neutralize, and target antigens. And you said before, for complement, I said I really don't care about all the entire domino effect of all that, but just realize we're going to neutralize receptors, and we're going to do something called opsonization, and that's kind of like we just kind of tag it with an antibody so that your T cells really can't miss it. Your T cells, they function in killing foreign cells, and I said before, these are hand-to-hand combat. Right? They're not going to send antibodies out. They're going to actually find that MHC1 marker that's antigenic. They've been programmed and introduced to formally. They're going to find it in any substance. Once they see it, they're going to lock on, and they're going to kill whatever that's attached to. I know I mentioned a lot about antigens, and I'm hoping that this is not foreign to all you, or maybe it's review. An antigen, or AG, sometimes you'll find it abbreviated, is a substance that provokes an immune response in specific lymphocytes. So anything can be an antigen. You know, it could be dust for myself, and we're not meeting in person, but if we were day one of class, I would tell you guys that you can't wear patchouli into class. If you're not familiar with that, it's an essential oil. I call it hippie juice, whatever, but I'm highly allergic to it. So that, for me, that's an antigen. And we'll talk about haptens here in a minute. All right, so these are antigens, but they're really, really tiny. They are so tiny that they don't provoke an immune response unless they're attached to another protein. And other proteins are huge. So once they attach to that, your immune system can't miss it. Right? So property of behaving as an antigen is called antigenicity. So it depends on, it's got to be foreign. Right? The size, we're going to talk about haptens. It's got to be big enough, 10,000 molecular molecule size. It's got to have that shape, all right, that's going to trigger that B cell antigen receptor. It's got to be accessible. It has to be in contact with your B cells, or they're not going to recognize it, right? All right. So characteristics of antigens. So anything that's foreign, all right, to be foreign enough, it's got to be perceived as foreign, not a normal constituent of the body. So it can't be something in your body that's normal, or your immune system was exposed to before and finds it normal. It's not going to trigger a response. Foreign cells and large complex molecules over 10,000 molecular weight are the most antigenic. They're so big that they are going to trigger a bigger response. All right. And here I said before, I remember I said before that some kind of antigens, or some kind of, some different types of foreign bodies can have more than one marker on their epitope. We're going to go over this here in a second. Antigen determination, or called an epitope. Epi means on, usually. Small molecular group that is recognized by lymphocytes. Antigen has many antigenic determinants. So you could have an antigen, and you can have all these different determinants on its epitopes, or on its site of that antigen. And theoretically, each one of these is going to represent a different antigen. And theoretically, you could have all of these antibodies in your body binding to that. Right? So how cool is that? That you could have one antigen of, let's say, you know, I'm allergic to cat hair too, but that's actually a hapten, because it's actually the saliva on the cat hair. But theoretically, if the saliva was on cat hair, and I breathed it in or somehow swallowed it or whatever, and let's just say that that cat hair or that saliva on there had 6 different epitopes on it, and each one of my B cells could recognize that from previous exposure, I could literally have 6 or 7 antibodies attaching to that. Your body can't miss that as a foreign substance. And in my case, it's going to send out all kinds of inflammatory responses, which can be not a good situation. All right, and here's the haptens. These are small foreign molecules that consist of only a determinant type. So they're tiny. They have the determinant type, but they're too tiny. Remember, they have to be 10,000 molecular weight. So this is literally like cat saliva. It's a hapten. So until it attaches to something larger, it's not big enough to trigger the epitope or trigger the antibody to produce it's not big enough to produce an antibody response. Not antigenic unless attached to a larger carrier. Carrier group contributes to the size of the complex meaning more than one thing, right? And enhances the orientation of the antigen. So it just makes it large enough that it will actually attach to that variable region and trigger the antibody production. So just some terms here. Allo antigens, these are cell surface markers and molecules that occur in some members of the same species, but not in others. Right? So we're all the same species, all right? So most of us may be allergic to some things, some of us may not, all right? So, you know, a good example of this would be peanut allergies or things that, you know, a certain percent of the population may be allergic to, and some of us may not be allergic to at all. Now, super antigens, we're talking about when we talk about some of these bacteria coming up. These are potent T cell stimulators, provoke an overwhelming response. So if I have a bacterium or something that can just literally produce super antigens and pump it out into the external environment, they come in contact with a T cell, any T cell, it's going to send the T cells into a frenzy, all right? And they'll just start attacking everything. It actually makes them go ballistic. An allergen is an antigen that evokes an allergic response, right? So you could have an antigen, if it doesn't evoke an allergic response in somebody, it's not an allergen, right? So autoantigens are molecules of self-tissues for which tolerance is inadequate. All right, and we're going to come up with this, we're going to come across this next chapter. We're talking about molecules of self-tissues for which tolerance is inadequate. And we're going to talk about later about some bacteria that are going to trigger an immune response, all right? And what's going to happen is once we produce antibodies to some of these bacterium, they're going to actually find that some of your tissues in your body after this response are going to trigger the same exact allergic response. And we'll talk about this with strep, all right, strep throat, and some of these things. So once your body has a reaction to certain of these streptococcal bacteria, it'll produce antibodies. Now these antibodies, unfortunately, are going to find the lining of your heart and heart vessels to look almost close enough to it that they'll actually start a response in your heart valves and heart tissue. So does that sound like a very good thing? Yeah. So that can be problematic. Right. Cooperation and immune reactions to antigens. The basis for most immune responses is the encounter between antigens and white blood cells. So that's usually what happens. We have to have the antigen, it has to trigger the white blood cells, right? And if the antigen doesn't trigger a specific white blood cell, a B cell or T cell, we're not going to have an immune response to it. So lymph node and spleen concentrate the antigens and circulate them so they will come in contact with lymphocytes. And we said before that there's a lot of afferent vessels coming into a lymph node or the lymph tissue, and there's only one efferent going out, and that's by design. We want this antigens to percolate in your lymph nodes so that we have a lot of exposure time. And we want to, hopefully, there's a B cell or T cell in that lymph node that has already been programmed for that specific antigen. So the B cells will make antibodies and the T cell will know the MHC1 marker on it and wage an attack on it. Okay. All right, so I want to get into the antigen antibody processing and presentation. Remember, the presentation is done by your dendritic cell, your antigen presenting cells. So once again, these are your macrophages, monocytes, your B cells, and sometimes your neutrophils. So T cell dependent antigens must be processed by phagocytes called antigen presenting cells. And once again, I said that these T cells are lethal, but they have to be formally introduced to the antigen. So an antigen could go right by a T cell. The T cell doesn't see it. It's got to be literally introduced to the antigen by a CD4 cell. Antigen presenting cells modify the antigen. I'm sure they consume them and process them. Then the antigen is moved to the antigen presenting cell surface and bound to an MHC receptor. All right. And try to remember it's an MHC2 receptor because I present it. All right. MHC1 receptors on all of your cells, unique to you or a twin, and only in your nucleated cells. So antigen presentation involves a direct collaboration among an antigen presenting cell and a T helper cell. And once again, your T helper cells, when you see that, you got to think CD4. Interleukin 1 is secreted by the antigen presenting cell to activate T helper cells. All right. So your antigen presenting cell, dendritic cell, monocyte, neutrophil, whatever it happens to be, produces interleukin 1. So just remember, it's starting the process. It's going to activate a T helper cell, your CD4 cell. And I said before, CD4 cells are what triage your entire immune system. Interleukin 2 is produced by the CD4 helper cell to activate B and other T cells. Right? So interleukin 1, first step, antigen presenting cell says, look, I found this wherever. Presents it to a CD4 cell. The CD4 cell says, yeah, you know what? That is foreign. I'm going to agree with you. It's going to trigger a response. All right. Then interleukin 2 is going to be produced by that CD4 cell. And it's going to say, listen, we've determined this is foreign. I need to activate all of the B cells to produce memory cells and plasma cells. So plasma cells are going to produce the antibodies. And I'm going to say, you know what? I'm going to get your cousin, the T cell, I'm going to present the antigen to him. So when he sees that same antigen on the MHC1 marker of all these foreign particles, remember I said it has to have a formal introduction. So we've introduced it to it. Now the T8 killer cell, all right, goes in and says, whenever I see that marker, I know that you're foreign and I'm going to attach to you. I'm going to lock on and we're going to destroy you. Hand-to-hand combat. Okay. So it's just kind of showing you the T helper activation. So this is your CD4. Remember, the dendritic cell has to present it to the CD4 cell. The CD4 cell has to agree it's foreign. All right. So this is your MHC2 marker. This is your antigen presenting cell here too. It's presenting it to, all right, your T helper cell. All right, this is your CD4 cell right here. All right, presenting it to the marker. Remember, T cells had there were the variable regions down here. They looked a little bit different than the B cell, but the CD4 cell MHC2 marker is presenting to your T cell. All right, it's formally introducing it there. Now the CD4 cell is the helper cell. It's going to triage everything. It's agreeing that this is foreign. All right. Now this arm literally swings out, locks onto here. They literally sign the contract, and we know that that CD4 is now activated. And your CD4 cell that triages everything, its next step is interleukin 1. Remember, to activate your B cells, and then interleukin 2 to activate your B cell memory cells, plasma cells, and your T cells. So your B cell responses. Usually your B cells are the first ones activated. Right? So B cell activation, antibody production. Once B cells process the antigen, they interact with the CD4 cell. They're stimulated by growth and differential factors. They enter the cell cycle in preparation for mitosis and clonal expansion. So what this is kind of saying here is once we activate that B cell, all right, it's going to start producing antibodies. All right, it's going to make more antibodies. It wants to make a bunch of memory cells, and it wants to make a bunch of clones. It wants to make a whole bunch of other cells and more plasma cells so it can produce antibodies if it needs to. Divisions give rise to plasma cells, and these secrete antibodies and memory cells that can react to the same antigen later. And once again, this is the theory behind all of our vaccinations, right? Or our prior exposures to whatever it happens to be. If you were exposed to something, if you're exposed to it again, you should have memory cells to it. All right, the only thing we can't really have vaccinations for, as far as I know for sure right now, are any RNA viruses, all right, because they mutate, all right? So influenza or any RNA virus, and unfortunately, I'm pretty sure the coronavirus is an RNA virus. So I'm not sure whether these vaccinations are going to work or not. Let's hope they do. All right. All right. So B cell activation. All right. We have our MHC2 receptor. Remember, these are your antigen presenting cells. All right. They're going to grab your microbe. They're going to present it to your B cell. They're going to lock and key. They're going to sign the contract right here. We see this. All right. Now, the B cell is saying, okay, I get it. I'm going to start making memory cells to it and plasma cells to make more antibodies. Now, we talked about antibodies before, but please realize these are the antibodies that attach to the B cell, and they have the antigen presenting region on them. But we also, if the B cell makes antibodies, it's going to literally take a bunch of these. It's going to pump them out into your bloodstream, all right, into your interstitial fluid in your bloodstream. So now they're floating all around. Whenever they come across that antigen anywhere in your body, they attach to it, they super tag it, they opsonize it, and now we have a great big complex molecule floating around with the antigen attached to it. Now your immune system is going to go and grab it, and now we can wage war to it, or your T cells can find that, attach to it, and literally kill it. All right. Here's your antigen antibody binding. So we just have to remember, this is the hyperavailable region on the antibody that binds to the antigen. And we said before a few times that it has to be a really good fit or it won't work, all right? So in truth, your immune system can make genetic general antibodies. They kind of, they're kind of common. They'll attach to things. It's not really junk antibodies, but it's something to neutralize it or draw attention to it. It's not an exact fit, but it's a close fit. Your book doesn't talk about that beyond the scope of the course, but that in truth, your immune system does it. But for this course, just kind of realize it has to be very specific. Right? All right, and here's what I was talking about before, and I mentioned last week. Principal antibody activity is to unite the antigen, to call attention to or neutralize the antigen for which it is formed. All right, so what we're doing is with opsonization, it's a process of coating microorganisms or other particles with specific antibodies so they are more readily recognized by phagocytes. All right, so we're going to super tag it. Right, we're attaching it to the molecule. All right, now your phagocytes, your macrophages and monocytes, they can't miss this thing. It's huge. It's like the USS Starship Enterprise. Right? It's huge. They can't miss it. Neutralization, the antibodies fill the surface receptors on a virus or the active site microbial enzyme. So let's say that your Staph aureus is secreting some kind of super antigen enzyme. We can send antibodies out to it, and we can plug up the receptors, right, to prevent it from attaching. Kind of, I mentioned on Tuesday, that's sort of like putting super glue in a lock. All right, or jamming up the code on a keypad. We can, we not be able to kill it, but we can at least jam up its mechanism a little bit. All right? In neutralization, we kind of go in and we just, like I said, we just fill up the receptor sites so it can't dock. So if we filled up all the receptor sites theoretically on a virus, or, you know, currently we're working, you know, we're talking about any kind of coronavirus, right? Because there's been others, SARS and other ones. If we could theoretically had antibodies to a previous, let's say you had a previous exposure to SARS or one of the other coronaviruses that were in the past, all right? If the antibodies were close enough, they could go in and jam up the receptors on that coronavirus. So even if it came in contact with your type 2 pneumocytes, it wouldn't be able to attach to your cells. All right, it can't dock on your alveolar cell, and it can't attach and get into those cells. So that would be good, right? All right, so agglutination, this should be familiar if you had A&P with me or other instructors. We should have talked about blood clotting or agglutination. All right, so antigen antibodies, the antibodies in blood from foreign blood coming into your body, it's going to recognize it as foreign. It's going to go in and cross-link it, antigen antibody reaction, and make agglutination. And that's a major problem because now we have great big complex molecules, all right, jamming up your capillary bed and potentially jamming up your glomerulus in your kidney, and that can be majorly not good. All right, complement fixation, activation of the classical complement pathway, right, and precipitation, aggregation of particle antigen. And we'll talk about this later in the course when we talk about RA and some of these other problems where we have precipitation of antigen antibody complexes and autoimmune diseases, right, or your body's attacking itself and parts of your body. And we have these antigen antibody complexes, and they start precipitating out of your bloodstream, and they start depositing in the vessels and other tissues. And then when your macrophages go to clean it up, they start causing major, major damage over and over and over again, causing joints, the synovial fluid and synovial membrane and the cartilage area to get disrupted. It can alter the shape of your joints and other things going on. Okay. Right. So function of your FC fragment, this is what actually binds to your cells and your macrophages, neutrophils, eosinophils, mast cells, basophils, and lymphocytes. So what they're saying is this fragment of the antibody is floating around. It found an antigen, attaches to it, it floats around, then it literally attaches to these specific cells. And if you remember what eosinophils do, or what mast cells or basophils do, once we attach to these, it's going to trigger that cell to produce whatever it produces. And we said before that mast cells, they release histamine, and basophils release histamine too. Remember, these are in your bloodstream, these are in your tissues. So once these attach to an antigen, they clump, then they go back and they find the receptor, the heavy region locks onto the specific cell and triggers that cell to release whatever that cell is designed to release. Right? It's going to binding of the FC region may cause release of cytokines. So it attaches to the cell, clicks on a switch, sends a signal to the nucleus. The nucleus starts producing whatever that cell is been waiting to produce, and the cell releases it through exocytosis. So let's talk a little bit about your IG or immunoglobulins. Yes, Robert. Is that what triggers your histamines? That's true, yes. Histamines on your mast cells or basophils. Yes. Right. So let's say that you were, Robert just asked me, is that what triggers your histamines? So let's say that you are stung by a bee or you have an allergic response to something. You're going to inhale that either through your respiratory system and your mucous membranes, or if you get stung by something, all right, that antigen from either the what you breathe in, the allergen that you're allergic to, or the bee sting is going to, the antigen is going to attach to these FC fragments, and they're going to literally antigen antibody reaction. Then it's going to take those clumps, especially your mast cells. Let's say you're stung, it's in your skin. Going to attach to that, it's going to attach to your mast cells, and your mast cells are going to release a whole bunch of histamine, bradykinin, a lot of inflammatory chemicals, and you're going to have like a histamine storm. All right. So here are your IGs, and I said before, these are your immunoglobulins. All right, so these are in your blood. These are proteins, and this is what we look for if you have, when we do blood titers. We want to see if any of these are elevated. We know something's going on. all right? So IgG is produced by plasma cells, and this is your primary response in memory cells. So initially, if you have an immune response or an antigen-antibody reaction, IgG generally is the first one produced, all right? It's primary response, or your first response, all right? It's going to bind to that and cause some kind of reaction. It's going to tag whatever that antigen is, all right? IG, I've mentioned this literally, I'm going to say 8 times so far. It's a monomer. Monomer means 1. Circulates in the blood. It's a dimer, all right, 2. And mucus and serous secretion. So once again, this is an immunoglobulin, IgA. Remember, it's in all of your mucous membranes. So anything that's exposed to outside your body has this. Right? We start talking about some of these sexually transmitted, I can't say disease, sexually transmitted infections. Some of these organisms are able to destroy IgA. All right, so now they're destroying the IgA. When they enter the tissue, we can't neutralize it or optimize it. We can't super tag it. All right, so this is another virulence factor for these organisms. They have a way of getting to the mucous membrane. We can't super tag them. And remember, right under your mucous membrane is your basement membrane. Underneath that is your capillary bed. So they go right into your bloodstream. Right. IgM, I said we're going to come across a great big one. It's got 5 monomers all attached. You're going to see it's huge. This one's huge. This is the first class synthesized following the IG encounter. All right, so let's say that you have an antigen encounter. All right, this is going to be the first one that's produced. All right, IgG is your primary response produced by your blood cells when you have a secondary, when you've been exposed before, all right? IgD is a monomer. It serves as a recognition receptor for antigen on the B cells. So all of your B cells are going to have IgD on them, all right? That's a question they love to ask too, all right? This is actually a receptor for antigen on all of your B cells. And IgE, we're going to talk about, is involved in allergic responses and parasitic worm infections, all right? So we talked about eosinophils before. And so I remember IgE, I just remember eosinophils because those are the white blood cells that are elevated when you have those parasitic worm infections. And I said last class, when you have allergic response, sometimes eosinophils will be triggered. Usually it's basophils or mast cells, but sometimes these can be triggered too, right? All right, so if you, let's say we did a blood titer and your IgE levels were elevated, all right, we would say, well, you know what, whatever's going on is either going to be an allergic response or it's going to be, there's probably some kind of parasitic worm infection because we checked your immunoglobulins in your blood and this is elevated. We know that whatever's triggering it is going to be allergic response or parasitic worm infection. Right. So this is your secondary response, all right, which is what we want. If we are exposed to something, okay, we want that secondary response to come right away. And we already said your B cells have already been triggered from your first exposure, right? So they have memory cells. They made a bunch of memory cells, and they have the recipe for the antibody. Right, so it's going to cause these plasma cells to start pumping out antibodies right away. You don't have to wait that long extended period of time to actually build up antibodies, and we don't have to wait for your B cell to actually be exposed to the antigen and trigger a reaction. It's already happened. We have the recipe on file already. Okay. So after second contact with the same antigen, all right, the immune system produces a more rapid, stronger response due to memory cells. It's called the amnesia effect. All right, so IgM is usually always circulating. All right, so our second exposure is right here. Right away, I said IgG is always excreted during your second exposure right away. So we have this great big general immunoglobulin to deal with it. All right. The secondary exposure, we don't have to wait anymore. We have the immediate response. And like I said, this is sort of the premise behind most vaccinations. All right. A rapid rise in antibody titer, mainly IgD, is sustained for several weeks. A smaller amount of IgM is also produced by naive B cells that have never been stimulated before. We stimulate them. And I said before in the beginning of class, we want this to happen because once we have a secondary exposure, we already have the recipe, our plasma cells are pumping it out, and I said before, they stick around for a very long time, making sure we've gotten or super tagged all of the antigens. All right, so this can be a week to 2 weeks. All right, and especially we can trigger our macrophages to stay on, and they literally, I said before, they're going to fight to the death until we wear them out and kill them, and then upon death they release this really cool like spiderweb net to trap everything around them. So if there's any bacteria or anything around them, they actually send out this sticky net to grab it so they can't go away. We sequester them for other macrophages to come along and kill. So cell-mediated immunity requires a direct involvement of T cells. All right, so whenever you see cell-mediated, remember, I know I'm droning on and on about this, but cell-mediated, I said your T cells are hand-to-hand combat, all right? So humoral or blood is always B cell and plasma cell. Cell-mediated is always T lymphocytes, right? Because they don't release anything. They literally go after it. And remember, these guys are have been formally introduced to the antigen, the MHC marker of the foreign substance. So once they recognize that, they recognize it, they attach, they literally swing this arm-like appendage out, they lock onto it, and then they punch a hole in it, and they excrete toxins into the organism to kill it. Right? T cells act directly with the antigen in foreign cells when presented in association with an MHC carrier. MHC1 is that antigen that you have that's unique to you, and most organisms out there have an MHC marker. T cells secrete cytokines that act on other cells. So they're going to attack, they're going to release cytokines. And remember, these are chemicals that they secrete to act on other cells. And we saw last week that these cytokines are in the interstitial fluid. So as the other cells are going through the blood, they literally are attracted to that chemical gradient, and they're going to go towards the concentration gradient. If you don't remember, it's in the last PowerPoint, and it's in the video I uploaded from the 21st for you guys. So sensitized T cells proliferate into long-lasting memory cells. So once we sensitize them, once we introduce them to the antigen with the CD4 cells, they also produce memory T cells. So we already have the code for the antigen. So if we ever get exposed to it again, they'll recognize it and they'll have hand-to-hand combat with it. Right? Once again, another component of a vaccination. We want this. It's a very good response. Right. So here's another chart for you guys. CD4, the helper cells, activates other CD4 cells and CD8 cells. Right. Remember, this is going to secrete the interleukin-2. That's going to activate your other CD4 and CD8 cells to make memory cells and your plasma cells, right, and clones. Right. Tumor necrosis factor, interferon. Right, we talked about that before. Responsible for delayed hypersensitivity. And we'll talk about that maybe not today because I'm losing my voice. All right, but delayed hypersensitivity reactions are only with T cells. So we're talking about hypersensitivity 1, like anaphylactic shock, all right, type 2, and type 3, which are all B cell, all right? So just kind of remember type 4 hypersensitivities are T cells, and that's usually when you have reaction to like poison ivy or some of these other things that you get exposed to it, and then a day or two later, your T cells start attacking your own tissue because of the superantigen and their response to the hapten or whatever you come in contact. Right? And this interacts with the MHC2 receptors. MHC2 receptors are those receptors from the antigen-presenting cells presenting that antigen. T helper cells, your CD4s, all right, these are derived from B cell proliferation, all right. And I said before, they're going to also have some of these activities. They dampen T helper 1 activity. So these actually go in, and I said they kind of put the brakes on overreactions if they're functioning. And then your T cell, CD8, remember these are your killer cells, cytotoxic. All right, cyto, like cytoplasm, toxic. So they literally go in, hand-to-hand combat. They'll kill anything that has that antigen on it that they've been presented to from the antigen-presenting cell. Destroy a target foreign cell by lysis. I said they go in, they attach, they lock on, they puncture a hole in it, and they secrete enzymes to destroy it. Important in destruction of cancer cells, virus-infected cells, all right? So it's going to cause the cell to produce apoptosis, and these T cells, if they see that antigen from the virus on that cell and they've been exposed to it before, they're going to notice that marker, or if the virus causes the outer membrane receptors to change, it's going to pick up on that. It's going to come along and say, you don't belong here. It goes in and just kills it, all right? Because it knows it's one of your cells, but it knows that the outer surface markers, the MHC1 markers have been altered. So it's going to go in, attach to it, and kill it because it knows that it was virally infected, and we don't want that cell to become a virus-producing factory. All right, and these are the cells that we're going to be talking about with graft rejection. All right, you know how your body's going to, if you get a graft or a bone marrow transplant, the transplant is going to have different markers on it. So if it thinks that the marker, if it's been exposed to those markers before, your antigen presenting cells present the marker of that graft as foreign. The CD8 cells are going to go in and destroy it. And, you know, we want to give them some immune suppressant drugs, and we can try to keep that at bay. All right. All right, so your T helper cells are the most prevalent type of T cells. Like I said, they regulate your immune reactions to antigens. They triage your entire immune system, including other T and B cells, also involved in activating macrophages and increasing phagocytosis. Cytotoxic T cells, cytotoxic, these are the killer ones. Remember, these are hand-to-hand combat. Destroy foreign or abnormal cells by secreting perforins that lyse cells. Perforins. So it's actually going to punch a hole in it and lyse the cells. Natural killer cells. So this was the one of the lymphocyte line that people tend to forget, and they don't really talk a whole lot about. It's not, it's crucially, I guess not important, but it's not as crucially as tested on. All right. These lack specificity. All right. They circulate through the spleen, blood, and lungs. And these, theoretically, they are able, they're T cells, but they're able to recognize pathogen-associated molecular patterns. So they're able to recognize anything that's not quite right. So if it's a cancer cell that the markers have changed on, or a virally infected cell, if we alter the outer antigens or MHC markers on a cell in our body, if it's altered at all, these guys can go along and notice it, and they go in and kill it. I was just saying, here's T cell activation differentiation. Here's your CD4 cell. Talked about it with B cells before, but it's going into, there's a MHC2 marker on a dendritic cell. It's activating your CD4 cell. And we said before, it's going to release interleukin, all right? It's going to cause the CD4 cell to be a memory cell now. It's going to produce other B cells, all right? It's also, we said it's going to activate more macrophages. It's going to send out those cytokines, right? Interferon, gamma. It's going to stimulate macrophages, because we know that these guys, if they're working, are going to create a bunch of damage, all right, dead cells. We need these guys to come along and clean it all up, all right? They're like little shop vacs that go and clean up everything, all right? And then we said before, interleukin-2 is released, and that's going to stimulate your CD8 cells, which are your natural killer cells. So here, antigen presenting cell is presenting it to your T cell receptors, right? They recognize it as foreign, they lock on it, they sign the contract, activates it that CD8 cell, it knows now to look for this antigen, right? It's going to find it on another cell. It's going to go in and destroy it, right? Or if it notices a cell that is damaged or has been altered by a virus or cancer, it's going to lock on and kill it, right? Even though it knows it's a normal cell, it's going to kill it. So it doesn't, if it's cancerous, it doesn't make more of itself, which are mutated, or if it's a viral infected cell, we want to kill it so it doesn't produce a whole bunch of viruses. Okay. Right. And here's literally a picture of a cytotoxic T cell attaching to a cancer cell. Right. Right. So we'll talk about T cells and superantigens more when we talk about Staph aureus and some of these virulence factors. So the reaction has drastic consequences. So this is called like a cytokine storm or cytotoxic storm. So this can cause major tissue damage. Superantigens are a form of virulence factor. I said before, virulence factors are something an organism can release, and it gives them a great big advantage. So if I can get all my T cells in the area to start attacking randomly all around the tissues, now my T cells are busy doing something else. All right, kind of what's going on in politics right now, right? So we're keeping everyone busy so we don't know what's going on behind the scenes, right? So provoke overwhelming immune responses by a large number of T cells, releases cytokines, causes blood vessel damage. We've all heard of toxic shock, right? It can cause major toxic shock and multi-organ damage. Right? We can start causing blood to clot inside of your vessels, and that will stop blood flow to your organs, all right, causing hypoxia, ischemia, and the organ tissue, the parenchyma will just start dying. Right. So we'll talk a little bit here about these. Should all be reviewed. Active immunity is what we really want. Results when a person is challenged by an antigen that stimulates production of antibodies by that B cell. B cell is naive. We find the specific antigen that unlocks the lock and key. That B cell gets activated, starts producing antibodies, memory cells. This takes a little time initially, but it's very long lasting. So we have active immunity. So if you were exposed to things as a child, hopefully you didn't get sick, but at least you knew that antigen was foreign. Your body made memory cells to it and plasma cells. So if you ever got the same antigen again, and it was a higher infective dose, your body would be able to take care of it immediately. We don't have to wait for that B cell activation, that naive cell. Passive immunity, so foreign antibodies are donated by an individual, does not create memory, acts immediately in short term. So if we have somebody who's exposed to something, all right, and we don't have time to, the host doesn't have time to produce antibodies. If we have antibodies around, we can give it to the person right away. So like snake bites, spider bites, they're saying that there's certain part of the population has antibodies to certain things out there, and they want that blood, they want the antibodies. So if we ever need it, we can directly IV it into the person, and they'll have the antibodies right away. All right. Natural immunity acquires as part of normal life experiences. All right. So if you were a kid, when I was a kid in the 60s or whatever, we ate dirt and paper, whatever. So we acquired immunity through that, hopefully. Artificial immunity acquired through a medical procedure such as vaccination. So that's the key. So we want to artificially give them part of the antigen, a capsule, attenuated viral particles, or whatever, to stimulate our active immunity. We're going to purposely give you the antigen that we know is antigenic, all right, so that you will stimulate production of antibodies. We're going to stimulate your B cells to that. Hopefully you have a B cell that can produce antibodies to that, all right, create memory cells, takes a long time and is lasting, all right. The only caveat I want to mention now, and you guys can Google or argue with me or whatever, is I don't really understand why we are vaccinating children before the age of 6 months, because your body can't your immune system isn't even up and running. It can't produce antigens. It can't produce antibodies, memory cells, or anything at that time period, as far as I understand. I've talked to several people about this. So anything before 6 months is questionable, in my humble opinion. Right. And there's just some examples of it from your book. Right. So immunization, manipulating immunity. So passive, where immune seroglobin, gamma globin, contains immunoglobulin extracted from pooled blood. So immunotherapy. So if somebody has an issue, we can take some of that serum with the antibodies and give it right to the person right away. Right. And that would be, something, like I said, with snake bite or spider bite or something that could be potentially very serious. We want to give them antibodies right away, so we can bind up that toxin. So we might not be able to get rid of it, but if we can bind up the majority of the toxin, we talked about neutralization. We want to go in and we want to neutralize all those receptors so that toxin can't go in and stimulate the deadly syndrome it can do. We want to neutralize it as soon as we can. Right. Artificial active immunity is we deliberately expose a person to a material that is antigenic but not pathogenic. So we want to give them part of the antigen, or we want to attenuate that virus so it's not viable or it won't become pathogenic, we hope, in the majority of cases. Right? Principle is to stimulate a primary, secondary immune response, what we saw before, that immediate response the second time you're exposed to it, to prepare the immune system for future exposure to a virulent pathogen. Response to a future exposure will be immediate, powerful, and sustained. And remember, I said, once we have a secondary exposure, that's going to stick around for a very long time. And you guys can read in your book, if you want, about how vaccinations are prepared. All right, so we want killed whole cells or inactive viruses. All right, we're hoping that we're not giving anyone any kind of activated viruses. The jury's out on that because, viruses are not technically alive. But we want something that won't be able to hijack your cells and replicate. And we want your body to make antibodies to that so we can bind it in the future so it won't be able to dock on your cells. Right, so we can use live attenuated cells or viruses. Or I said before, we can take antigenic molecules derived from bacteria, cells, or viruses. So we can just take part of the outer surface of a capsule or a slime layer, or something that would normally be recognized by your B cells or your antigen presenting cells. We take a part of that and we put it in the body, and we hope that your dendritic cells, your antigen presenting cells, will grab a hold of that and present it to your CD4 cells for recognition and stimulate your B cells and T cells against it. We have memory cells, plasma cells for B cells, and memory T cells already in the arsenal. So if it ever actually comes across the actual bacteria or virus, we already have antibodies to it right away. We don't have to wait for that titer to build. And then we, genetically engineered microbes or microbial agents. So there's qualities of an effective vaccine. We definitely don't want it to cause the actual pathogen. We don't want it to cause the actual disease, with measles or whatever it happens to be. I'm not going to get into the anti-vaxxer or the the autism argument with anyone. So that's that's something you guys can research on your own. Just showing you here killed or inactivated viruses. We're just going to take parts of it, the actual parts of it, and we want it to distribute that in the body. And like I said, we really want our dendritic cells or antigen presenting cells to pick up just that portion of it. We don't, the rest of it's, theoretically dead. It can't come back to life. The viral particle can't replicate. We've killed everything inside it, but we just want the markers. So, theoretically, if we could find the antigens on coronavirus, doesn't even have to be the one currently, part of that, and we can inject it into somebody, and your body will make antibodies to that portion of it then we theoretically will have antibodies to something similar to that, or close enough to it, to neutralize it, so it won't be able to attach to any of the host cells that it might be able to get into otherwise. Right? I'm showing you live attenuated cells or viruses. Okay. Yeah, I think that's it. And these are just antigenic particles. So we're just saying we could take just parts of it, what we want. So if we're talking about bacteria, we're going to take just part of the capsule, pneumococcus or meningiococcus for meningitis or pneumonia, anthrax, hepatitis B, all right, some of the exotoxins from diphtheria, all right, that causes major damage in the back of the throat and the respiratory system, tetanus. We just want to take part of that, and we want a capsule. We're not taking the whole virus. So hopefully your immune system will recognize that if you actually came in contact with any of this, it would automatically make an antibody to it immediately. And I think that's all I wanted to cover on this. Yeah, here's a list. This should be scary, of all the current vaccinations they give someone. It's frightening, but anyway. Okay. So I think that's it. You guys can study up on herd immunity. You know, the jury's out on that. My opinion, I'm going to keep to myself. But if you have a vaccination and it works, then whether someone's vaccinated or not, you should have immunity to it or whatever. Okay, so immune individuals will not harbor it, reducing the occurrence of pathogens. So we have herd immunity. We want that in the majority of the population. Less likely that a non-immunized person will encounter the pathogen. But please realize, I'm just going to say that some part of the population can't be vaccinated, all right? So if they're immune compromised or elderly, or, in my humble opinion, if they're under 6 months of age, they really shouldn't be vaccinated. So we want the rest of the population to have herd immunity to do it, so that not everyone theoretically is walking around with the disease. Right? All right, so I guess we'll wrap it up here, because this picture scares me too, the amount of vaccinations that they give people. So I will look, I guess I'll look forward to seeing everyone Monday. So do we have a consensus on WebEx or Zoom? Anyone? Okay. If you guys can hear me. Bob says we're still muted. Shit. All right. Okay, so trying to mute again. I didn't do anything. Okay. All right, so I'm going to fix the mute setting before Monday, but I'm going to go back into Zoom. All right, so I just want to do Zoom because it's more private. All right, I really would prefer that I keep the recordings myself. I'll give them to you guys on Blackboard, but I really don't want my intellectual property out there. I know that sounds crazy, but that's just my opinion. So I'm going to do Zoom, or if I can get into Loom, we should try Loom. Right? So I will fix the mute problem for Monday. So I'm going to start at 5, but I'll assume there's going to be an issue or two. So we'll start at like 5:05. All right, everyone, have a good weekend. If you guys need more time for the test, I realize there was a breach and it's due tonight. Let me know. In the interim, I will be posting the exam for Unit 2. So I think what I'm going to do is give the majority of you all the same questions. And instead of 8 random questions, I'll give you guys half of the questions. You'll all get the same question. And then I'll just randomly generate like 4 questions rather than 8. All right, so just to be a little bit more fair. All right. So if you guys have any questions, email me. I know that you're muted for some strange reason. Or you guys can text me 716-228-5565. Just please don't text me after 9 p.m. because I'll be sleeping. Don't text me before 6. All right. All right, so everyone, thank you for joining. I will try to get all the bugs out. I hope I didn't overwhelm everyone. Please review the video. If I made a mistake, let me know. We can maybe go in and edit it. It's the other thing with Zoom, I can go in and edit, and I can word over everything. So if you have any questions, I know you guys are muted, text me and I'll answer them as soon as I can. So have a good weekend, and I will see you guys Monday. All right, good night. Thanks, Bob, for everything. I couldn't do it without you. All right. Thanks.